right? So here the phenotypic ratio of F2 generation is deviated significantly from F2 phenotypic ratio of Mendel's dihybrid cross in peas. In Mendel, what is the phenotypic ratio in dihybrid cross? 9 is to 3 is to 3 is to 1. What is the genotypic ratio? 1 is to 2 is to 2 is to 4, 1 is to 2, 1 is to 2 is to 1. That is the genotypic ratio. But whatever the Morgan had got here result, the result is different from Mendel dihybrid cross, right? In F2 generation, the ratio is different. Whatever crossed he taken as per Mendel only, there is different types of the characters, one dominant, one recessive. Again F1 crossed, procedure same but result is different. He found that Morgan found that 98.7 of F2 progeny are parental and 1.3 of F2 progeny are recombinates. This point you should remember carefully. This point here you can observe carefully. Morgan found the result in F2 generation 98.7 are parental and 1.3 of F2 progeny are recombinates. So what you have got it from this point? Most of the progenies are similar to the parents. 98.7% means maximum percentage similar to the parents. Progenies are formed. Only very less, 1.3 progenies only recombinant, new varieties are formed. That means what? Recombination very less takes place. Very less recombination takes place in this scenario. Why very less recombination? The reason is what? The reason is we discussed earlier. The genes are closely presented, tightly linked. Whenever genes are tightly linked, there is no crossing over. Crossing over is very minute less. So that only both the genes are reaches to the same gamete. Whenever both the genes are reaches to the same gamete, then obviously similar to the parent characters only formed. And how much recombination is formed? 1.3%. Whatever the percentages is the most important, you should remember an objective point of view. So in cross A, Morgan got 98.7 parental characters, 1.3 recombinant characters progenies he got in F2 generation. Cross A, what he had taken? Yellow bodied and white eyed female recessive with brown body and red eyed male dominant. Clear? Right. Okay. So let us see the cross B. In cross B, what he had taken? He crossed white eyed miniature winged female, white eyed miniature winged female. That is what? That is recessive. And wild red eyed, the wild red eyed and normal winged male, that is dominant. So here we taken here, we discussed wing size normal is dominant, miniature is recessive, eye color red eye is dominant, white eye is recessive. So here white eyed miniature winged female, that is recessive character, with wild red eyed normal winged male, that is dominant. Earlier body color and eye color taken. In this cross, eye color and wings size taken. You got it? Right. So in F1 generation, you have got F1 generation in the form of red eyed normal winged female, same. Red eyed normal winged female he got, because sexes are changed in F1 generation because of it is X linked. And white eyed miniature winged he got. white-eyed miniature winged male he got. Female he taken parent, he got male as a progeny in F1 generation. Okay, sexes only changed, whatever the combinations are not changed in F1. So what about F2? He intercrossed the F1 hybrids and obtained F2 generation. So the phenotypic ratio of F2 generation is deviated significantly from F2 phenotypic ratio of Mendel dihybrid cross in piece. Here also same. The result is different compared with the Mendel experiments. He found that in F2 generation, what he had found, 62.8% of F2 progeny are parental and 37.2% of F2 progeny are recombinants. Compare with cross A, in cross B recombinants increased, parental progenies are decreased, right? In F2 generation, cross B, what percentage Morgan got? Progenies 62.8% of parental, 37.2% of recombinants. In earlier, 98.7 parental and recombinants are 1.3 only, but here recombinants percentage is increased. Whenever recombinants percentage was increased, means what is the reason? Crossing over more takes place. In what case more crossing over takes place? The genes are tightly presented or loosely presented? The genes are loosely presented, distantly presented, more distance. The distance between genes are more. Whenever the distance between genes are more, then weak linkage, then crossing over takes place, so that only recombinants. So both genes are separated. Those genes are reaches into the separate gametes. So more recombinants, less parental. More means more recombinant, less parental means what? Compare with cross A, I am telling, right? So in cross A, 98.7% parental, 1.3 recombinants. In cross B, 62.8% parental and 37.2% recombinants formed. In both of the crosses A and B, he observed that the two genes did not segregate independently of each other and F2 ratio is deviated. In both the crosses A and B, he observed that the two genes did not segregate independently. The gene segregation independently was not takes place of each other and the F2 generation is deviated. F2 ratio is deviated. Suppose independent assortment was takes place as per Mendel, then the result also same. Here the result is different. Morgan observed that recombinant types were low, 1.3% in cross A as compared to cross B. Correct or not? In cross B, recombinants are 37.2. In cross A, recombinants are 1.3. It means genes for white-eyed and yellow body were tightly linked. Obviously, what it means? It means genes for white eye and yellow body were tightly linked. Whenever tightly linked is there, then only there is less recombinants are formed, 1.3. Genes for white eye and miniature wing were loosely linked. Genes for white eye and miniature wing were loosely linked, so that only high recombination shows, 32.7% shows. By this phenomena, what you had understood? By this phenomena, on chromosomes, some of the genes are tightly linked, some of the genes are loosely linked. Which genes are tightly linked? That genes exhibits in progenies very less recombination. Which genes are loosely linked? That genes are exhibits high recombination, high recombinants, high recombination and high recombinants are produced. You got it or not? Right, up to this clear? So once again I am revising. I will show the diagram, you can understood easily, right? Okay, Morgan dihybrid cross in Drosophila. Morgan, Thomas Hunt Morgan done the experiments in Drosophila and expressed the concept of linkage and recombination.